All right, welcome back. We're going to do a quick overview of Chapter 7 of Campbell's Biology, membrane structure and function. So Concept 7.1, cellular membranes are fluid mosaics of lipids and proteins. So here are the phospholipids. Remember that they are amphipathic, so they have hydrophobic regions that exclude water and hydrophilic regions which interact with water. Now, remember that although I oftentimes describe this as a droplet of oil, I want you to remember that these hydrophobic heads do actually interact with water and form hydrogen bonds. This means that the water interacting with these hydrophilic heads strengthens this overall structure. So the water itself, its tension, can help keep this together. So it's a little bit stronger than just a droplet of water. Now, this phospholipid bilayer acts as a boundary between two aqueous compartments, and for the cell, it separates the outside of a living cell from the inside of it. Now, this works because plasma membranes exhibit selective permeability. So some things can pass through while other things cannot. Here is an image of a phospholipid bilayer, a more molecular structure. So here, all these red and gray things are water molecules. The green tails here are the hydrophobic lipids, and these yellow balls or spheres are the phospholipid heads. So this in the green is all one molecule. Here in the background, we actually have a transmembrane protein passing through. Let's bring this to life and take a look at it. You can see that while water is moving around, it is not passing through the membrane. Now, we describe the membrane as a fluid mosaic model. And this means that the individual lipids or phospholipids within this bilayer can move around and acts as a fluid. But let's not confuse that with the idea that the proteins are just randomly distributed in the membrane. A vast majority of the proteins are actually both connected to the extracellular matrix and the cytoskeleton, making their placement on the surface of the cell very intentional. Now, that being said, when we start thinking about the actual phospholipids themselves, they do tend to move fairly freely. So here we have a mouse cell dyed red, we have a human cell dyed blue. If we were to fuse that together, those phospholipids would mix and mingle, and within an hour, it would be fairly evenly distributed. Now, this is true for some proteins in that they, with the lipids, can drift laterally, but this is not true for most proteins. Most of the proteins are anchored in some way to either the intracellular matrix or the extracellular matrix or the cytoskeleton. Now, as temperatures cool, membranes will switch from a fluid state to more of a solid state. Okay, just think about butter and think about butter melting. The temperature at which this switch occurs depends completely on the lipids. So when you have a membrane that is rich in these unsaturated fatty acids that have kinks in their tails, you can't pack tightly, it tends to be more liquid at a cool temperature, to where when you have rich saturated fatty acids, this would tend to be more viscous. In order for membranes to actually work properly for a cell, they need to be about as fluid as solid as salad oil. So we want it to be fluid, but not just completely liquid. Now, there are also these things called cholesterol, and we often demonize cholesterol as the thing that gives you heart attacks, but cholesterol is actually a major part of our membranes and a necessary part. These steroid molecules are hydrophobic in nature, most of it, with one little exception up here. And so they can fit into the membrane, and they actually moderate the fluidity. And this works differently at different temperatures. So in warm temperatures, these cholesterol molecules actually restrain the movement of the phospholipids. So consequently, at warm temperatures, it keeps it more solid. While at cool temperatures, it actually prevents these phospholipids from forming van der Waal interactions and keeps it a little bit more fluid. So cholesterol works both ways. It keeps it both more fluid and more solid than it should be otherwise. Now, within this membrane itself, we also have various proteins attached. When we have a protein that is adhered to the membrane in some way, shape, or form, we call that a peripheral protein. When we have a protein that actually extends into the phospholipid bilayer, we call that an integral membrane protein. It can do this because it has hydrophobic regions or nonpolar regions. Now, we can also have particular integral membrane proteins that actually go completely through the membrane. We call these transmembrane proteins. And if you were to think about this, the regions of the protein that interact with the hydrophobic must be stretches of nonpolar amino acids, and they would oftentimes take the form of a helix. Helix are by far the most common structure to pass through a membrane. Now, the activity of the different proteins that are in the membrane kind of determines what that membrane's specific function is. And there are 6 major functions of membrane proteins. One is transport, so to specifically allow things through that wouldn't otherwise be able to go through. It could be an enzymatic protein, so it changes one thing from one thing into another, carries out a reaction. By putting it in the membrane, we can localize it to a particular region. In this way, we can organize a series of reactions together. Here you can see this comes in, turns into a square, then goes into here, then turns into a circle, right? So we can link things together in a membrane. They can also act as signaling molecules. So we see something like a hormone coming in from the outside, it activates this protein, and it sends this signal and causes the cell to do something. This could be involved in cell-to-cell recognition. So we have our cells that link together. So it can either be through this recognition mechanism or through a very specific intercellular joining, which is very tight and specific. These proteins also interact with both the cytoskeleton, or there are proteins that specifically interact with the cytoskeleton and others that interact with the extracellular matrix. This kind of sandwiches the membrane between these two more rigid barriers and helps organize the overall structure. Now, something to consider is membranes do have distinct insides and outside faces. So here we can see these lipid bilayers. One layer is painted blue, one is painted red. And the inside and outside is intentionally architected by the cell in order to achieve particular functions. So here, if you look at the inside of the ER lumen, that is the same surface as the inside of a vesicle. It is analogous to the inside of the Golgi apparatus and is ultimately analogous to the outside of the cell. So by putting things into the ER, when we discussed that you could make proteins in the ER or in the cytoplasm, by putting it into the ER, you can ensure that it goes to particular locations. Or if you were to insert things into the membrane, say it's only on one side of the membrane, we can ensure that it gets put onto the outside to interact with the outside world, or that it's put into a vesicle to be secreted. Just recognize that there are phases. So, cellular membranes are fluid mosaics of lipids and proteins. Remember, these molecules are amphipathic. They can move around in space. They can move laterally with each other. This lateral movement is dependent on the temperature and the types of lipids that are in there. Cholesterol can help moderate that fluidity. We do want it to be semi-fluid. But this membrane does not exist by itself. It exists with all of these proteins, peripheral proteins, integral membrane proteins, actual transmembrane proteins that go all the way through. Some of these proteins have various different types of functions, connecting cells together or allowing things to pass through, enzymes, signaling molecules, but they also connect to the cytoskeleton in the extracellular matrix, trapping the membrane in between. So the lipids may be able to move between, but a lot of the proteins are in fixed positions. And these membranes definitely have faces associated with it, and the inside or the luminal space of all these um, organoids are analogous to the outside of the cell. So concept 7.2 is membrane structure results in selective permeability. So when you have something lipid-like, it can pass straight through the membrane because it's not going to be excluded from this hydrophobic region. Non-lipid molecules would tend to not pass through. Now, the actual reason is the water is actually holding on to it and not allowing it to pass through, to where the water is actively pushing the stuff towards this region. Now, this allows plasma membranes to be selectively permeable, so hydrophobic things can go through, and hydrophilic things can not easily go through. Now, in order to facilitate the transport of things that couldn't ordinarily go through, we need to have these transport proteins. Some of these transport proteins are called channel proteins and can just allow things to pass straight through. They're usually specific for a particular molecule. For example, we have something called aquaporin, which allows water molecules to pass through fairly freely. This allows water to actually pass through the membrane, but not allow larger things to go through. We also have other specific transport proteins that can move even larger things through, like maybe an ion or a particular sugar. So the selective permeability of this membrane is what allows us to separate the outside world in a very controlled manner. So we can allow particular things to pass through because the water is actually pushing lipid-like things towards the membrane and it doesn't really resist coming back out. Non-lipid molecules would be held by the water and prevent it from going through. So in order to allow particular things to come through, we have to have these membrane channel proteins to allow things to go through. And there are the first two concepts from chapter 7.